We have seen in part 1 that humoral immunity is provided by proteins known as antibodies. Antibodies are proteins belonging to globulin fraction of plasma proteins, and since they perform an immune function, they are also known as immunoglobulins. An immunoglobulin molecule or antibody molecule is made up of two types of polypeptide chains, which are known as light chains and heavy chains. The light chains have a variable region and a constant region. Heavy chains also have a variable region and a constant region. The variable regions of heavy chains and light chains come together and they form an antigen recognition site which can recognize a particular antigen. The antigen may be a free molecule or it may be a component of some cell like a bacterial cell, or it can be a component of virus or some other microorganism. And then we had seen that these immunoglobulins, depending upon the type of heavy chain that they possess, can be classified into five classes: IgA, IgD, IgE, IgG, and IgM. The different classes of immunoglobulins differ in their concentration, their half-lives, and also in their activity. Immunoglobulin G is present in the highest concentration out of the five classes of immunoglobulins. Its concentration in plasma is 700 to 1500 milligrams per deciliter. It has also got the longest half-life out of the five classes. The half-life is 23 days. IgG is active against bacteria as well as viruses. The second highest in concentration is IgA. The plasma level of IgA varies from 60 to 500 milligrams per deciliter. Its half-life is much shorter, 5 to 6 days. It is strongly antiviral and weakly antibacterial. The concentration of IgM is 40 to 200 milligrams per deciliter. Its half-life again is 5 to 6 days. It is strongly antibacterial and weakly antiviral. The remaining two classes, IgE and IgD, are present in very minute concentrations in plasma, and therefore they are known as minor immunoglobulins. Their half-lives are 2 to 3 days each. IgE is mainly anti-parasitic, but it also mediates allergic reactions. The function of IgD is not known. This is the basic structure of immunoglobulins, which we have seen earlier. The molecule is made up of two light chains and two heavy chains. The light chain can be divided into a variable region and a constant region. Similarly, the heavy chain also can be divided into a variable region and a constant region. And as we have seen earlier, the variable regions of light chains and heavy chains form the antigen binding site, which recognizes and binds an antigen, and then the constant region of the heavy chain performs the effector function. The purpose of effector function is to prevent the antigen or the pathogen from exerting its harmful effects. The basic structure of the immunoglobulin molecule can exist in the form of a number of variants, and these variants include idiotypes, isotypes, and allotypes. As we have seen earlier, the variable region of light chain and heavy chain which are present in an immunoglobulin molecule form the antigen recognition site, and this antigen recognition site has got a typical three-dimensional structure which is complementary to the epitope or antigenic determinant of a particular antigen. And this typical antigen recognition structure is known as, or it constitutes, an idiotype. So every antigen recognition structure present in different immunoglobulin molecules constitutes one idiotype. We have two antibody molecules here. The similarities in colors and differences in colors represent the similarities in amino acid sequence and differences in amino acid sequence. So in the two molecules that we have here, the constant regions are of the same color. That means the amino acid sequences of the constant region are identical. But the variable regions are shown in different colors. That means the amino acid sequences in the variable regions of these two molecules are different. So here we have two molecules belonging to the same class, having the same constant region, but having different antigen recognition sites. So these two molecules will identify and bind two different antigens. So each of these variable regions, the antigen recognition sites, constitute an idiotype. So we have two different idiotypes here. We have got hundreds of thousands of antibody molecules having different antigen recognition sites, different antigen recognition structures. So each of them is known as an idiotype. Another variant is known as isotype. We have seen that the immunoglobulins are divided into five classes: IgA, IgD, IgE, IgG, and IgM. And IgA is subdivided into IgA1 and IgA2. It has two subclasses. IgG has four subclasses, which are IgG1, IgG2, IgG3, and IgG4. Each class and each subclass constitutes an isotype. The classes and the subclasses constitute different isotypes. Each class is an isotype, each subclass is an isotype. So we have seen that immunoglobulin G exists in the form of IgG1 or IgG2 or IgG3 or IgG4. So these are four different isotypes. Immunoglobulin A exists as IgA1 and IgA2. So these are two isotypes. IgD is one isotype, IgE is one isotype, and IgM is one isotype. Now the differences in the isotypes, different isotypes, lie in the constant region, the amino acid sequence of the constant region of the heavy chains. So we have two antibody molecules here. As we can see, the variable regions of these two molecules, light chains as well as heavy chains, are of the same color. That means the amino acid sequence of the variable region of light chain and heavy chain in one molecule is identical with the amino acid sequence of the corresponding part of the second molecule. So these two molecules have an identical antigen recognition site. They will recognize the same antigen, they will bind the same antigen. But their constant regions, the constant regions of the heavy chains are different. They have got different amino acid sequences. So this means that these two molecules belong to two different classes or two different subclasses, but they recognize the same antigen. So these two molecules are two different isotypes because they belong to two different classes or subclasses. The third type of variants are known as allotypes. So these allotypes are antibodies or immunoglobulins of the same class which have a very slight difference in the amino acid sequence in the constant region. The difference is only of one or two amino acids. These allotypes are formed by mutations, point mutations, changing one or two amino acids in the constant region. And these allotypes are inherited from the parents. We have two antibody molecules here. The variable regions are identical. The constant regions are almost identical, but there is a very small difference here and here in the amino acid sequence. And this small difference means these two are two different allotypes. Now, our immune system can synthesize hundreds of thousands of different antibodies. And each of these antibodies has got a different antigen recognition site. So they can recognize different antigens, they can find different antigens. And the diversity of antibodies is so much that the antibodies that we possess can recognize practically every antigen that we may encounter. Actual antigens as well as the potential antigens. For example, the synthetic chemicals which are not yet synthesized, which may be synthesized in future, but our antibodies are ready to recognize even such chemicals, such antigens which do not exist as yet. So this diversity is huge. We have seen that every antibody has got a unique antigen binding site which is complementary to the epitope of a particular antigen. And this specificity in antigen recognition is because of a unique amino acid sequence in the variable region of light chain and heavy chain. So in every antibody molecule, the light chain has got a unique amino acid sequence. The heavy chain also has got a unique amino acid sequence in the variable region. And we also know that the amino acid sequences of all the proteins are encoded in the genes which are present in our DNA. So one gene encodes the amino acid sequence of one protein. If we can synthesize millions of different antibodies which have unique antigen recognition structures, antigen recognition sites, this means that there must be a gene for every individual antibody. So to synthesize millions of unique antibodies, we need millions of different genes. But this is not possible. The total number of genes in human beings is just about 30,000. And out of these, less than 200 are antibody genes. So we have a problem how we can synthesize millions of different unique antibodies from less than 200 genes. The reason for antibody diversity or the molecular basis of antibody diversity remained an enigma for a long time. And in the end, it was Tonigawa who solved this mystery. He showed us that antibody diversity arises from rearrangement of genes. That means the 200 or less than 200 antibody genes that we possess can be rearranged in different ways to form a huge range, a vast range of different antibodies. Genes for light chains and heavy chains are present on different chromosomes. In genomic DNA, the genes for light chains and the genes for heavy chains are not complete genes. They are split genes. The gene is split into certain segments. And when a new B lymphocytes, antibodies are synthesized by B lymphocytes or B cells. So when a new B cell or B lymphocyte is being formed, the segments are brought together to form the complete gene for a light chain and the complete gene for a heavy chain. As far as light chains are concerned, the light chain gene is split into three segments. So instead of a complete gene present at one place in the DNA, there are three segments which constitute the light chain gene, and these segments are present at different locations. And these segments are known as variable segment or V segment, joining segment or J segment, and constant segment or C segment. So a complete gene will be formed when a variable segment, a joining segment, and a constant segment come together. Now, genes for these three segments, V, J, and C segments, are present in three different clusters. There is a V cluster, there is a J cluster, and there is a C cluster. And these three different clusters are present on two different chromosomes. We have seen that the light chains can be of kappa type and they can be of lambda type. The genes for kappa light chains are present on chromosome 2 and the genes for lambda light chains are present on chromosome 22. But these genes for kappa chains, the genes for lambda chains, are present in three different clusters: the V cluster, the J cluster, and the C cluster. Now, for kappa light chains, there are 40 genes in the V cluster. There are 5 different genes in the J cluster, and there is a single gene in the C cluster. For lambda light chains, there are 30 genes in the V cluster. There are 4 genes in the J cluster, and there are 4 genes in the C cluster. The V segment and the J segment encode the variable region of the light chains, and the C segment encodes the constant region of the light chain. So when a new B cell is being formed, one of the genes in the V cluster joins one of the genes in the J cluster, and the intervening DNA, the DNA in between, is deleted. So one V gene and one J gene are joined together, and this is known as VJ joining. So two gene segments, one V segment and one J segment have been joined, and the intervening DNA has been deleted. And then this VJ combination plus a C segment gene will be transcribed. The product will be HNRNA. And then this HNRNA will be processed, it will be modified to form the messenger RNA for a light chain. And this messenger RNA will be translated, a light chain will be synthesized, the light chain of the immunoglobulin. We see an example of light chain gene rearrangement here. It is for kappa type of light chain. There are 3 different gene clusters. This is V cluster having genes starting from V1 ending with V40. Then there is this second cluster, J cluster, starting with J1 and ending with J5, and then the third cluster is the C cluster, in which actually there is only one gene. When gene rearrangement occurs, one of the V segments joins one of the J segments. So in this particular B lymphocyte, this V2 gene in the V cluster is going to join J5 gene in the J cluster. So the DNA in between V2 and J5 is deleted, and V2 and J5 have been brought close to each other, and this gene rearrangement is known as VJ joining. And then there is only one gene in the C cluster, that is C kappa. So this V2J5 combination will be transcribed along with C kappa, and the hnRNA which will be formed will be spliced to form the messenger RNA for light chain. And this messenger RNA, this is the part derived from V2, this is the part derived from J5, and this is the part derived from C kappa. So this V2 and J5 are going to form the variable region of light chain, and C kappa is going to form the constant region of light chain when this messenger RNA is translated. For kappa light chains, there are 40 genes in the V cluster and 5 genes in the J cluster. So when one of the genes in the V cluster joins one of the genes in the J cluster, the possible number of combinations are 40 multiplied by 5. That means 200 different VJ combinations can be formed when one V segment gene combines with one J segment gene. We have 30 genes for V segment and 4 genes for J segment for the lambda light chains. So when one V segment gene combines with one J segment genes, the possible combinations are 30 multiplied by 4. So 120 different VJ combinations can be formed in case of lambda light chain. And as we have seen, this V and J segments encode the variable region. So 120 different variable regions of lambda light chains can be formed. But in a given B cell, there is only one particular VJ combination. So only one particular variable region. When we come to the heavy chains, the gene is split into 4 segments. And these segments are variable segment or V segment, diversity segment or D segment, joining segment or J segment, and constant segment or C segment. The genes for heavy chains are present in 4 different clusters. So we have 4 segments, and each segment is present in a different clusters. So 4 different clusters for heavy chain genes are present, and they are present on chromosome 14. Now out of these 4 segments, the B segment, the D segment, and the J segment encode the variable region of the heavy chain. The C segment encodes the constant region of the heavy. Now for heavy chains, there are 40 genes in the B cluster. There are 25 genes in the D cluster. There are 6 genes in the J cluster, and there are 9 genes in the C cluster. Now, when a new B cell is being formed, one of the D segment genes, one gene in the D cluster, will join one gene in the J cluster. The intervening DNA, the DNA in between, will be deleted. And this gene rearrangement is known as DJ joining. That means one D segment has joined one J segment. Then one of the genes in the V cluster joins the DJ combination, and again the intervening DNA is deleted. And this process is known as VDJ joining. So now we have got a combination of one V segment, one D segment, and one J segment. And these three V, D, and J segments encode the variable region of the heavy chain. Now this VDG combination, which has been formed in a particular cell, will be transcribed along with the first gene in the C cluster. And the first gene in the C cluster is C mu. So VDG combination and this C mu will be transcribed. A heterogeneous nuclear RNA, hnRNA, will be synthesized. Then this hnRNA will be modified, it will be processed to form the messenger RNA for heavy chain, and this messenger RNA will be translated to synthesize the heavy chain. So in this messenger RNA, the VDJ combination encodes the variable region. And we have seen that the number of genes in V cluster, D cluster, and J cluster are 40, 25, and 6 respectively. So the possible number of combinations, VDJ combinations, are 40 multiplied by 25 multiplied by 6, that is 6,000. So 6,000 different VDJ combinations can be formed by gene rearrangement. But a given B cell has only one particular VDJ combination, and therefore only one particular amino acid sequence in the variable region. These are the 4 clusters of genes from which a complete gene for a heavy chain will be formed. There is this V cluster, D cluster, J cluster, and C cluster. There are 40 genes in the V cluster, V1, V2, V3, etc. up to V40. There are 25 genes in the D cluster, D1, D2, D3, etc. There are 6 genes in the J cluster, and there are 9 genes in the C cluster. When a new B lymphocyte is being formed, the first gene rearrangement that occurs is DJ joining. In this rearrangement, one of the genes from the D cluster will join one of the genes from the J cluster. The intervening DNA will be deleted. For example, in this particular lymphocyte, it is a D2 gene from the D cluster which has joined J6 gene from the J cluster. So DJ joining has occurred, D2 and J6 have been brought together, and the intervening DNA has been deleted. Now second gene rearrangement will occur, in which one of the genes from the V cluster will be joined to the DJ combination which has already been formed. For example, in this particular lymphocyte, it is the V1 gene from the V cluster which will join the DJ combination. So after this combination, V1, D2 and J6 have been brought together, and this is known as VDJ joining. Again, the DNA intervening between V cluster and D cluster has been deleted. So now we have this VDJ combination which will encode the variable region of the heavy chain. And then we have this first gene in the C cluster, which is C mu. And this mu type of heavy chain is present in IgM. So when this VDJ combination and the C mu are transcribed, and HN RNA will be formed, and the HN RNA is then modified to form the messenger RNA for heavy chain. So this messenger RNA for a particular heavy chain has this V segment derived from V cluster, this gene derived from D cluster, this J gene derived from J cluster, and C mu. V, D, and J will encode the variable region, C mu will encode the constant region. In this way, hundreds of different light chain genes can be formed by rearrangement of genes, and each of these light chains will have a unique amino acid sequence in the variable region. Several thousand heavy chains can be formed which will differ in the amino acid sequence in the variable region. So each will have a unique amino acid sequence. So hundreds of different combinations of light chains, thousands of different combinations of heavy chains. And when the light chains combine with the heavy chains, the number of combinations that are possible are in millions. So the variable region of all these antibody molecules will be different from each other. They will be complementary to different epitopes of different antigens. So each combination will be specific for one particular antigen. So this is how we can form a huge number of different antibodies, each of which is specific for one particular antigen. But in a given B cell, a given B lymphocyte, there is only one combination. So this B cell will synthesize only one particular antibody which can recognize one particular antigen. This diversity, which occurs because of gene rearrangement, or which occurs because of variable combination of different gene segments, is known as combinatorial diversity, and this is the major mechanism by which antibody diversity arises. And it has been calculated that in human beings, nearly 2 million different variable regions can be formed by combinatorial diversity. When gene rearrangement occurs, a part of DNA has to be deleted, different fragments have to be joined, and for this we require some enzymes. The enzymes which take part in gene rearrangement reactions are the DNA repair enzymes plus two enzymes known as RAG1 and RAG2. RAG1 and RAG2 are encoded by two genes which are known as recombination activating gene 1 and recombination activating gene 2. While combinatorial diversity is the major mechanism for antibody diversity, this is not the only source of diversity. There is a second source of diversity, which is known as junctional diversity. When gene rearrangement reactions are taking place, when different gene segments are being joined, some of the nucleotides may be deleted from the ends, and some nucleotides may be added to the ends. And this addition or deletion of nucleotides creates new base sequences. And because of new base sequence, the amino acid sequence will also change. And these addition or deletion reactions, again, they require some enzymes. The nucleotides are deleted by exonucleases, and nucleotides are added to the ends by terminal deoxyribonucleotide transferase or TDT. And then there is a third and final source of diversity, which is somatic hypermutation. When a B cell has been formed and it encounters an antigen, mature B cell encounters an antigen, a number of point mutations can occur in the variable region of the gene. Because of point mutation, bases are substituted. And when bases are substituted, amino acid sequences are changed. So this results in further diversity. So the antibody diversity arises from combinatorial diversity, junctional diversity, and somatic hypermutation. So the complete genes for light chains and heavy chains are formed by rearrangement of genes. Now, if we look at the complete gene for a heavy chain, it has got different regions, different segments. Every gene is preceded by a promoter. So the heavy chain gene is also preceded by a promoter site or a promoter region. And just upstream of this promoter region, there is a regulator also. In addition, an enhancer element is present within the gene. It is located between the VDJ combination and the C segments. There are 9 genes in the C cluster. So we have 9 C segments, and each C segment is preceded by a common sequence, a repetitive short sequence, which is known as a switch sequence. The promoter for the heavy chain is weak, but the expression of the heavy chain gene is increased by the enhancer element, which is located within the gene. So here we have a complete heavy chain gene starting with a regulator. Then there is a promoter, VDJ combination, enhancer element, and then a switch sequence, and every switch sequence is followed by a constant region gene. There are 9 C segments in the heavy chain gene. These are C mu, C delta, C gamma 3, C gamma 1, C alpha 1, C gamma 2, C gamma 4, C epsilon, and C alpha 2. And in between each of these, there is a switch sequence. Among the 9 C segments, the first is C mu. So after VDJ joining has occurred, this VDJ segment is transcribed along with the first constant segment, which is C mu. And the mu type of heavy chain is present in IgM. So IgM is the first immunoglobulin to be formed upon entry of any antigen. We have seen that antibodies perform different effector functions to deal with the antigen. The effector functions include neutralization, opsonization, complement activation, etc. And different classes of immunoglobulins perform different effector functions. For example, IgM mainly acts by complement activation. IgA acts mainly by neutralization. IgG can perform all the effector functions. When we encounter an antigen for the first time, we have seen that IgM is the first immunoglobulin to be formed. But IgM acts only by complement activation. So we may need a different effector function to deal with the same antigen. And for this, we require immunoglobulin of a different class but recognizing the same antigen. So this new immunoglobulin, which will replace IgM, should recognize the same antigen, but it should perform a different effector function. For this, the VDJ complex, which has already been formed, which is already present in the DNA of the B lymphocyte, should join a downstream constant segment gene instead of C-mu. Normally, this VDJ complex is transcribed along with C-mu. But when we require the immunoglobulin of a different class, the same VDJ complex should join a downstream constant segment gene and the intervening DNA is deleted. So now we will have a different constant segment. So an immunoglobulin of a different class will be synthesized. But since the VDG complex is the same, the antigen specificity will remain unchanged. So the new immunoglobulin molecule will be of a different class, but it will recognize the same antigen. And this is known as class switching or isotype switching. That is switching from one class to another or from one isotype to another to perform a different effector function against the same antigen. This is an example showing class switching from IgM to IgA2. That means the first antibody to be synthesized was IgM, but now we are going to switch to IgA2. These are the 9 genes for constant segment starting with C mu. It is followed by delta, then gamma 3, gamma 1, alpha 1, gamma 2, gamma 4, epsilon, and alpha 2. So the BDJ complex, which is already present in the gene, is normally followed by C mu and is transcribed along with C mu. So the antibody to be formed is IgM. But when class switching is required, the DNA is looped and the VDJ complex is brought close to some downstream constant segment gene, in this case alpha 2. So this VDJ complex joins C alpha 2, the intervening DNA is deleted. So now the gene that we have has got this VDJ complex followed by C alpha 2. So the antibody which will be synthesized will be IgA2. But since the VDJ complex is unchanged, the antigen recognition will not change. The new antibody will recognize the same antigen but will perform a different effector function. And we will continue from here onwards in part 3.